Hi, I'm Sophia, and today I'm going to be discussing some of my work on how fidelity, proximity, and climate factors contribute to flannelmouth sucker use of tributaries for spawning. Work by Kevin Bestrin and Kevin Thompson's groups on the Gunnison and White Rivers in Colorado, McIver's work on the Paria River in Arizona, and Bauer's work on Wyoming, Colorado River tributaries has demonstrated that flannelmouth sucker throughout the Colorado River Basin move into tributaries for spawning. Although the flannelmouth sucker has experienced range reduction from historical distributions, they remain numerous in many rivers, including the San Juan River. With continued changes in flow regimes and climate change, it's important to learn what we can from these native fish. This is in comparison to some other native suckers, like the razorback sucker, that no longer naturally recruit in the river, where we have to draw conclusions from hatchery-reared populations or similar species like the flannelmouth sucker that may or may not be useful proxies depending upon the question. Additionally, suckers hybridize, so understanding reproductive ecology is important for conservation efforts of multiple species. Drought has led to fewer available tributaries, and those that do exist can be easily damaged by water extraction, pollution, or fragmentation. These habitats provide different conditions than those available in the main stem, which can be particularly important in our dammed systems that no longer experience flooding-induced heterogeneity or connection to the floodplain. And again, we know that fish use these tributaries either for spawning or for refuge during high flow events. And Nate Cathcart, a former student in our lab, showed just that, where the bars on this graph represent detections, and we see a majority of the detections occurring during spring spawning season, with a few additional detections during high flow events in the summer monsoon season. He found that annual patterns in flannelmouth sucker detections related to temperature, flow, as well as seasonal effects. So my question is really looking at what makes a flannelmouth sucker decide to move into these tributaries for spawning. So what are the controls, and how does spawning in tributaries contribute to the overall population? And we anticipate things like habitat variables being important for the decision to move into tributaries. Tributaries could have different substrate size or embeddedness, as well as a difference in low velocity habitat for larval suckers. Biotic interactions, such as predation risks or interactions with conspecifics, could also contribute to a fish's decision to move into a tributary for spawning. Resource availability might also be different between tributary and mainstem habitats. Individual fish might experience these factors differently depending upon how far they are from a tributary during a certain time of year, or if they've previously spawned in this tributary, or if it's their natal ground. Additionally, abiotic environmental factors such as temperature or discharge of both tributary and mainstem habitat could contribute not only to if a sucker spawns in these tributaries, but when it moves into them. So I'm going to start by looking at some of these individual factors and asking the questions, what is the return pattern of Mcilmore Creek spawning fish, and where do fish spawning in Mcilmore Creek come from? So again, I'm looking at Mcilmore Creek, which is a tributary in the middle reaches of the San Juan River and is mostly perennial as a product of irrigation and water development associated with the creation of McPhee Reservoir. And it's really one of the only perennial tributaries, substituting out for some other tributaries that have recently experienced low flow events. Several thousand flannelmouth have been tagged in the San Juan River with passive integrated transponders, or PIT tags, and an in-stream permanent antenna located near the mouth of Mcilmore Creek allows us to get detections throughout the year that we can then pair with habitat or hydrologic variables to start examining some of these patterns of spawning migrations. So jumping into some of the data, looking at some of the fish that Cathcart had tagged, during each 2013 shown here in orange and 2014 shown here in blue, about 750 spawning adults were tagged in Mcilmore Creek. And looking at these fish now over the past 9 years, we see that during most years there was a decrease in the number of detections, but we still have a relatively high number of these fish that are being detected on an annual basis. Roughly 200 fish from each of these years is still detected in Mcilmore Creek every year. So looking at individual fish detection history, this diagram is just going to show us the top row representing individuals that were tagged in 2013 and not detected in 2014, or tagged in 2013 and detected in 2014 as the bottom row. And we'll continue to add years here. And the right-hand column represents the number of individuals out of 750 that display each of the 8 most common return histories. And what we see is that it's most common for a fish to be tagged and never seen again, but that in general, fish return in consecutive years, with fish being detected every single year since 2013 actually being the 4th most common. So our next question was, where do the fish spawning in Mcilmore Creek come from? And to answer that, we pit tagged 314 individuals along the San Juan River in December of 2020. And here we have a graph of a logistic regression where the x-axis represents distance from Mcilmore Creek where these fish were tagged, and the y-axis represents the probability of use, or whether a fish moved into Mcilmore Creek in the spring for spawning. And we see that more than half the fish tagged near Mcilmore Creek were also detected in Mcilmore during the spawning season, and that this number decreases as you move further from the tributary. We also implanted 45 Flannelmouth Sucker with radio transmitters at this time. And the reason that can be beneficial is with pit tags, just because it wasn't detected doesn't mean the fish wasn't there. But for each of these radio transmitted fish, we have specific locations during the spawning season. So we know that of the 15 fish implanted with radio transmitters near Mcilmore Creek, 60% of them moved into the tributary for spawning. And of those tagged further away, 14% moved into the tributary. So a similar pattern to what we saw with the pit tag data. Next, we'll talk about some of the abiotic environmental factors and determine if there's interannual variation in the timing of fish moving into Mcilmore Creek and how this variation relates to environmental factors. So here we have January through May on the x-axis, and each one of these curves represents the number of detections on a given day during a year. So in most years, we see this 2-humped pattern indicating when fish arrived and when they left. And in certain years, we see that this is quite different. So in 2017 and 2019, fish arrived roughly 25 days earlier than the other years. And that translated also to fish staying longer. So if we looked at individuals' arrival and departure date, they actually spent more time in Mcilmore Creek as well. And here we've paired this with a hydrograph, again looking at January to May and each year on the y-axis. And what we see is during 2017 and 2019, during these years where fish arrived earlier, there was also higher discharge earlier in the year. The years like 2021, 2020, and 2018 that have fairly low base flow also showed similar patterns of when fish arrived and left. So some association between the hydrology and when fish are showing up, but I'm hoping to look more into that in the future. So kind of the take-homes here, we did see relatively high site fidelity with fish returning on an annual basis for spawning in Mcilmore Creek. We also saw that fish that were tagged during the winter closer to Mcilmore Creek were more likely to use it for spawning, and that over half the fish we tagged within the couple kilometers of Mcilmore did indeed move into there for spawning. Finally, we did see interannual variability of when fish showed up and how long they stayed in Mcilmore Creek during the spawning season. And it does appear that there is some correlation between the hydrologic regime and when fish appeared. So in general, my research is hoping to examine how flannelmouth suckers may be using tributaries to maintain relatively high abundances, how environmental factors influence these patterns, and think about how they might change with changing flow patterns in the future. So some of my plans moving forward are to look at that main stem movement, again, using that radio transmitter information. We have almost a year now worth of data. So looking at where these adult fish are going during the rest of the year. Considering habitat features of where these adult fish are during the rest of the year, but also thinking about how habitat varies between tributary and main stem and where some of those radio transmitted fish were during the spawning season if they were not in Mcilmore Creek. And the next big component to think about is the contribution to the population. So if fish spawning in Mcilmore Creek is really only half the story, there might also be differences in survival of larval fish that are spawned in tributaries versus the main stem. And so we're hoping to use some genetic analysis to really get at this question of if there's differential survival and what the contribution of these tributary spawning fish is to the population as a whole. And since we're not able to do this in person, I'd love for you to contact me with any questions or thoughts about this study. And with that, I'd like to acknowledge all the help I've received so far doing this work. Thank you.